Welcome to the Bible and Science. This is Dave Prentice. I'm a retired public high school teacher, certified in all branches of science and math. I've also taught on the Bible and Science for many years, both in the U.S. and on many overseas mission trips. This is episode 33 of the Bible and Science. We've been dealing with some of the microscopic aspects of DNA, about how it reproduces, what it produces, how it goes about producing proteins and so forth. Now we're going to shift a little bit and move toward the visible effects of DNA, that is, the macroscopic level. As I've pointed out before, Michael Behe's book, Darwin's Black Box, gives some wonderful examples of arguments for design. And the best one, the most powerful one, is called irreducible complexity. This would be something illustrated by a mousetrap, for instance. It has to have a certain minimum number of pieces. It's got to have a hammer, a trigger, a latch, a spring, and a base at the very minimum. If it has any less than that, it's not going to work. So it has to have at least a certain amount. This is what he calls irreducible complexity. We're going to see a number of things that are irreducibly complex in living things. If any part is missing or not working properly, the whole thing is just a useless waste of material. Last time I touched on the way the antibody systems works. When an invader comes into your body and other vertebrates, they will encounter antibodies. These are little molecules that can bind to over 10 billion distinct shapes, because there are 10 billion, at least 10 billion possible combinations of amino acids in the heavy and light chains, which don't take the whole DNA. They just take 4 small segments of the DNA. The antibody gene clusters, number 1, 2, 3, and 4, allow the cells to produce 10 billion distinct types of antibodies. But I also want you to realize it's not enough to have the antibodies themselves. Where did they come from? They came from the cellular mechanism, and then once they're there, all they do is mark as warning flags that there's an invader that needs to be destroyed. Then there are other systems, the lymphocytes, NK lymphocytes, T and B lymphocytes, that will then attack and destroy any invaders that those antibodies are attached to. So all of these parts have to be there in order for the system to work. That is, it's irreducibly complex. So a multicellular organism would quickly die without an immune system. But before the very first multicellular organism evolved, there would have been no way to have an immune system, because it's produced as a result of DNA, which wouldn't have been present. So far, we've only been considering the effects of mutations on single cells. But on a larger scale, from one major type to another major type, could mutations force living things to evolve from one major type to another major type? For instance, what would need to happen if some of the oldest known plants, such as stromatolites, which were already performing photosynthesis, in order for them to evolve to the animal kingdom? Remember, the other argument that I consider is extremely strong for design is extreme improbability in a specific pattern. And Dr. Behe used the example of trying to get a groundhog across a 1,000-lane superhighway. Well, I'm looking for extreme improbability. You wouldn't say it's absolutely impossible. You just say, for all practical purposes, that would never happen. Okay, so in order for something to evolve from the plant kingdom to the animal kingdom, or the plant domain to the animal domain, many members of the animal kingdom need elements that they cannot get from the soil. copper, manganese, selenium, iron, calcium, potassium, and so forth. They have no way to get these directly from the environment. But lucky for us, many plants furnish exactly the materials that the animals need in a usable form. So every living thing is supposed to have come from the same single-celled ancestor, LUCA, the last universal common ancestor. There would have had to be two complementary and parallel series of mutations going on for hundreds of millions of years. The first thing we would consider an animal, its DNA had to mutate in a way to require those specific minerals, those specific elements. But then the plant DNA would also have to mutate in such a way to go get those minerals out of the ground in order for them to be available to the animals. You and I can't just go eat a handful of dirt and get the elements we need. The plants put it into a form that our bodies can use. So is this an amazing series of millions of coincidences, or could it perhaps point toward design instead? There are a few other, well, there are many, many, many other difficulties of one major type evolving into another major type. What would be necessary? You and I have got roughly 3 billion base pairs, ACGT and so forth, in our DNA. They work in pairs. So bacteria, which supposedly would be somehow akin to us way back when, billions of years ago, bacteria have only a few million base pairs in their DNA. So if everything evolved from one single-celled ancestor, then the most highly evolved things, which is actually not humans, but we're among them, we ought to have the greatest number of base pairs. So if you look at simple bacteria, they have 1 to 2 million base pairs, reptiles 1 to 2 billion. Most plants have anywhere from hundreds of millions to several billion, but sharks and frogs have more than we humans do. We have about 3 billion, they have about 4 billion. There's some varieties of beans that have 30 billion base pairs, and there are even some varieties of lilies that have up to 100 billion base pairs. So when Jesus was asked about needing things, why did he pick the lilies? He said, consider the lilies of the field. They neither toil nor spin, but yet Solomon in all of his glory was not arrayed like them. Lilies have some of the highest numbers of base pairs in their DNA out of anything. But if you look on that chart, show me where's the gradual evolution from bacteria to insects to birds, reptiles, and so forth. It's nowhere to be seen in DNA. Medical science has learned how to use specific viruses to treat mutated genes. So a virus starts out containing a small amount of DNA or RNA, and its normal genetic contents are chemically removed in gene therapy, and then a desired unmutated gene is inserted. If everything goes according to plan, the virus then puts the corrected gene into the patient's cell. Well, that's fantastic technology that scientists have developed, but it's irrelevant to evolution. Gene therapy goes exactly contrary to nature. It's anything but random. It requires a great deal of intelligent design and careful control to insert the specific desired genes into a cell. It's not the virus that helps the species. It's the artificially inserted gene that the technicians and the scientists decided, let's put this in there. So the viruses have to be carefully selected to be effective on a particular species without harming that species, without harming the subjects. And the original mutation doesn't improve anything. The gene therapy is an attempt to undo it. Evolution supposedly works by the accumulation of mutation. We're trying to remove the mutation by inserting the normal unmutated gene. So speaking of viruses and gene therapy and so forth, well, bacteria are some of the most common life forms. There's so many of them because they're so small. The vast majority of bacteria are not harmful. They work as decomposers. This makes them crucial to life on Earth. And speaking of viruses, only a small number of viruses affect any given species. Most viruses are not harmful. They're essential to the survival of bacteria. They act somewhat like bees pollinating flowers. This allows bacteria to exchange genetic information that will help them to survive in the event of a change in their environment. And besides that, viruses are insufficient to add billions of nucleotides to the DNA. Remember, the simplest life forms supposedly would have just a few million base pairs, nucleotides, but the most complex ones, including us and lilies, have billions or hundreds of billions of nucleotides. So could a virus gradually, or an accumulation of viruses, gradually add billions of nucleotides? There are only a few dozen viruses that affect any given species. Each virus usually contains only 1 to 3 genes. So if a species gained dozens of genes this way, and the process repeated hundreds of times, the number of nucleotides added would not be hundreds of billions, it would be perhaps a few million. And the process would stop once multi-celled organisms evolved. Viruses are usually not passed on to future generations in sexual reproduction. Well, what about if existing genes were duplicated? Mutations don't add genetic information. Viruses add only a small amount. So where did all of these extra DNA genes in the DNA in the higher organisms come from? Well, many evolutionists are grasping at straws, and they believe there must have been duplicate copies of existing fully functional genes that were occasionally added during reproduction. And then the new extra copies mutated to produce new features. Well, adding extra copies of genes is not usually beneficial. This is what causes Down syndrome, Warkani-Patau, and Edwards syndromes, which are not good conditions that you want to inherit or give to your children. So the extra copies don't do a whole lot of good. And then the duplicate gene already contains instructions to perform a specific function. If you change it through mutations, this is going to cause a loss of genetic information, not a gain. Mutations would have to be responsible for the very first appearance of blood, heart, lungs, bones, eyes, ears, legs, wings, every other structure not found in the simplest organisms like bacteria. Meanwhile, remember the mutations in the animal kingdom have been making animals depend upon a whole bunch of extra elements off the periodic table that they can't use right out of the soil. But meanwhile, mutations in the plant kingdom happen to cause the plants to go out and grab those exact elements to make them available for the members of the animal kingdom that were going to meet needing them. Well, the whole purpose is to get rid of God. We're going to explain it by all-powerful mutations instead. Maybe you've heard of superbugs, bacteria that acquire immunity to certain antibiotics and so forth. Well, here's my illustration. Suppose you have an automated barnyard set up to feed and water 1,000 chickens. 950 of the chickens are white, 50 are brown. One of your business competitors sneaks in, puts poison into the food and water supply. But it turns out only the white chickens are susceptible, and they all die. So the machinery sweeps away the carcasses, and after a short time, all you have left are brown chickens. There's no competition. So the 50 brown chickens breed and multiply quickly. You should really go check your hatchery more often than my illustration. But the 50 brown chickens breed and multiply quickly, and any white babies die quickly. So what do you have before long? 1,000 brown chickens. You started with brown and white chickens. You ended with brown chickens only. Has anything new been added? No, this is not evolution. This is elimination of certain genes, the genes that would have allowed the appearance of the white chickens. Well, a similar thing can happen with bacteria. We introduce antibiotics, and from the very beginning, many of them are susceptible. However, a few of the bacteria are immune to those antibiotics from the beginning. There are only a certain number of antibiotics out there right now, and if the manufacturers would make some new ones, then that would take care of the so-called superbugs. But we've got a few bacteria that are immune to the existing antibiotics from the beginning. Since there's no competition, the other bacteria have died out, the resistant strain multiplies quickly, and it fills the ecological niche. And it's an interesting thing that even in samples of bacteria that have been frozen for centuries, we always find that some are resistant to any given antibiotic. We dig up stuff from the Arctic Circle or Antarctica, and some of the bacteria are immune to any antibiotic you come up with. They've always had the resistance. So has anything evolved? Is this a case of evolution? No, not at all. Evolution requires the addition of new genes, new features that weren't there before. This is not the addition of anything new. This is the elimination of something that was there already. Here's another thing that I was unaware of. It's called pleiotropy. There's only a small part of the DNA that contains the instructions, the coding instructions necessary to produce proteins. On the coding sections, it was long thought that one gene gave you one protein. Well, we've discovered no, it's a lot more complicated than that. But experimenters have deliberately induced mutations in many genes in higher organisms. They want to know what would happen if I do this. And to their surprise, almost every gene that's been studied in this way has affected not just one, but two or more body structures. So we must conclude, I write computer programs, I do subroutines. We must conclude that many of the segments of DNA are used like subroutines in a computer program. They're used two or more times. For instance, if I wrote a program and I tell the program, watch for a key press, I didn't tell it which key right there. It could be any key on the keyboard. It looks for a key press, then it branches off according to what key was pressed. Well, likewise, many segments of DNA seem to be used two or more times. So DNA contains far more information, and it's encoded in a far more complex way than we ever realized. This goes back to the probability. What is the probability that this is just an accident? If you've got it in your mind that there could not possibly be any sort of an intelligent designer, then it doesn't matter what anybody shows you. Your mind is made up. I shouldn't confuse you with the facts. But if you're open to the possibility, oh, there could be some sort of a design in nature, I think you'd have to recognize, yeah, there surely is. There's a segment of DNA, or several segments of DNA that used to be called pseudogenes, which means false genes. And you'll frequently see these referred to as junk DNA. Well, cells are made of tens of thousands of different types of proteins. Each protein is made of hundreds of copies of the same 20 amino acids, but arranged in different sequences. So for many years, we've known that a small number of the segments of DNA contain the coding to produce specific proteins, and the saying was one gene, one protein. Yeah, and many evolutionists complained that much of the rest of DNA didn't do anything, but it was junk DNA left over from an earlier stage of evolution. So since the genes didn't have a known function, they were called pseudogenes, that is, false genes. No, they're not junk DNA at all. A cell is composed of a great many proteins. Well, first you have to put together the proteins correctly. And we dealt with that in the last episode about how the genetic code leads to the production of this amino acid and this amino acid, and then how they come together and how they're folded correctly into proteins and so forth. So there are a great many proteins in a cell. They have to be assembled correctly. Now, suppose you're building a house. You can order all of the raw materials needed to build a house: the nails, screws, roofing shingles, wires, paint, electrical outlets, wood, glass, light fixtures, and so forth. Now that you've got the pieces there, they were delivered, now you have to assemble it. You need building plans. You can't just have a pile of junk there, even if it's potentially useful junk. You need building plans, and then you need skilled workers. Scientists working with data from the Human Genome Project have discovered hundreds of thousands of what are called all LNCRNAs, long non-coding RNAs. They perform the regulatory functions necessary to put the parts together into a cell. They don't have any visible proteins being produced, but still they make sure that the cell does what it needs to do. So these segments, which are actually most of the DNA, the majority of it, are no longer considered pseudogenes. They're regulatory genes. So there are many species that have similar segments of DNA that have no known function. Well, the evolutionists who claim they're pseudogenes, junk DNA, have the attitude that we are so smart, if we don't know what something does, it must not do anything. Well, that's ridiculous. We only know a tiny percentage of what happens in DNA. We've already learned that some of the segments thought to be useless, including several of the pseudogenes, have a definite function after all. Some pseudogenes are thought to be involved in cell differentiation. As a member of the human species, you have heart cells, lung cells, kidney cells, liver cells. They all have the same DNA, but in one cell, what switches on turns it into a heart cell, and another one it turns it into a brain cell, and so forth. So some of the genes, so-called pseudogenes, have now been found to have a regulatory function that turns other genes on and off. And then DNA is a three-dimensional structure. It loops back on itself in many places. Pseudogenes may be involved with furnishing the structural information, saying, okay, loop back and touch here and fasten there. Then there are also some segments of the DNA that contain coding information that's not obvious because the genes are non-continuous, as I talked about previously, on the part of the DNA that codes for antibodies. It's 4 different locations on the DNA. And if you didn't know, you would look at it and say, well, they're not connected in any way. Yes, they are. They produce the antibodies. So it's also possible, if damage has crept into the DNA, many of the so-called pseudogenes may be damaged copies of formerly functional alleles. That's not evolution. That's deterioration. And then something you will frequently see is that the protein sequence of cytochrome C shows evolution. Bacteria have a certain sequence in it. Jawless vertebrates have a certain sequence. Vertebrates have certain sequence. Yeah, so out of thousands of families of protein, the only one that can be used to support evolution is cytochrome C. For any given family of proteins, the percent difference between any 2 major groups it's not linear, it's hierarchical. For instance, variation between any bacteria and any insect will be about the same. Any insect, any vertebrate about the same. Any bony fish and any mammal about the same. Humans, anytime a monkey will be about the same. It's not an evolutionary pattern. It's sort of a mosaic pattern, a hierarchical pattern. And then what's called the second pillar of evolution is homology. It's the presence of similar structures in otherwise dissimilar creatures. For instance, many land vertebrates have 4 limbs. So if those similarities show evolution from a common ancestor, then homologous structures should be produced by homologous genes. They're not. Genes that produce similar structures seldom, if ever, occur at corresponding locations on the DNA of the affected animals and plants. So I've mentioned before Dr. Michael Behe's book, Darwin's Black Box. I want to strongly recommend that to you. The expression of black box is used in science occasionally, where something goes in, a bunch of factors, and something comes out. So it doesn't matter what's happening on the inside. We call it a black box. Well, in Darwin's day, the cell was considered more or less like a black box. So Dr. Behe's book makes reference to that. And I want to say he's not a biblical creationist like I am. He's a theistic evolutionist, but his book is wonderful. There are some other homology problems, similar structures in dissimilar types of creatures. Not only do they come from dissimilar genes, they often come from different body segments in the embryo. For instance, in insects, the adult organs can come from totally different parts of the body. In butterflies and ants and bees, new structures develop only as old ones are replaced. In beetles, the foregut and hindgut develop without any cell destruction. In plant seeds, most biologists consider the seeds of conifers like pine cones homologous, similar to those of flowering plants, angiosperms, but key parts of the seed form in ways that Michael Denton says profoundly differ in the two groups in a number of important respects. In vertebrates, there's a different sequence of development depending upon what you're looking at. The gastrula stage, the blastula stage, the alimentary canal, the forelimbs, they develop from different embryonic segments. The kidney, ureter, amniotic, allantoic membranes, they develop differently in different species, even though they're all supposedly descended from a common ancestor. And then the third pillar of evolution is called biogeography. This is where the same species or genera of animals and plants are sometimes found in different places throughout the world, but there are variations. So this phenomenon arises because of distance or natural barriers, not because of evolution. It has nothing to do with new features evolving. Creation can account for biogeography just as well as evolution. We believe that the first members of every particular group, every major type of creature, had all of the genes necessary, and later they would diversify. So the genes were there, but you didn't see the visible results. Those didn't come along, or might not have come along for a long time, but the genes were there from the beginning. Well, prove that. I'm not going to try. Prove that this is not what happens. Biogeography is something you can't prove either. So let me give you an example of how genes might become sorted out. Suppose the beak shape is determined just by 2 alleles, big B dominant, which gives a straight beak, and little b recessive, which gives a curved beak. So if both parents have both alleles, big B, little b, then both will have straight beaks. But it's possible they could combine in a number of ways during breeding. If the father and mother each produce at least one big bee, you're going to get a straight beak. But if they both furnishes only the little bee, then the baby will have a curved beak. So on average, about 1 in 4 babies will have a curved beak. This is the kind of thing that Gregor Mendel started writing about at the time of Darwin. So let's give an example, a made-up story, of course, how specialization could produce distinct groups. Suppose there's an island 50 miles long and a mile wide. It's got a dormant volcano in the middle. It contains curved beak and straight beak birds, trees suitable for feeding both varieties. So on average, 1 out of 4 has a curved beak. But then one day the volcano erupts. The poison gases blow to the east, kills all of the birds and all the trees capable of feeding straight-beaked birds. For some reason, and this can happen in nature, the other trees on the eastern half are more resistant to the gases and they survive. Okay, things settle down. The volcano quits erupting. The birds on the west begin to spread to the east again. Those with straight beaks find no food and they either go back or die. Those with the curved beaks would thrive on the eastern half of the island, not on the western half. They no longer have the gene for straight beaks, and before long the eastern half of the island is populated only by curved beaks. And then another disaster strikes sometime later. A great windstorm blows many of the birds to another island. It's too far for them to fly back. The trees on the other island are only suitable for feeding straight beaks. So any curved beaks that are blown over there, they're going to starve to death. So before too long, there's no food on the island suitable for curved beaks. They all starve. And soon the new island is populated only by straight beaks. So we now have 3 distinct groups that all came from one original founder population. The founder population has a mixture. About 3/4 have the straight beaks. And then the curved beaks on the eastern side, they all have curved beaks because there's no food for straight beaks. And on the other island, all you have are straight beaks because there's no food for curved beaks. Look at evolution going on. It's not evolution. Nothing new evolved. The groups became specialized as the genes were eliminated. You can certainly tell that this topic can go on, and it will continue with the next episode. We will continue talking about the effects of DNA at the visible macroscopic level for many more episodes.